Good morning, everyone. How are you today? Today we'll talk about meiosis or meiotic. We can we can say reduction division. It occurs in living organisms that reproduce by gametes. And gametes here depends on the type of the living organism. There are gametes in humans and animals, and there are another gametes in flowering plants. First of all, in humans and animals. In humans and animals, this division occurs in the testis, which is meiosis cell division, to produce the male gamete, which is sperm, and in the ovary to form the female gametes, which is ovum. So, testis produce male gamete, which is sperm, and ovary produces female gamete, which is ovum. Plural of ovum is ova. Plural of sperm is sperms. But in case of flowering plant, it occurs in anther, which produces male gamete, which is pollen grain, and ovary to produce egg, which is female gamete. Each produced cell contains half number of chromosomes of the parent cell. So, for example, this is anther that produces, which is a reproductive cell that produces male gamete, which is pollen grain, and this is ovary, which produces ovum or egg, which is the female gamete. In the other side, we have testis in human and animal that produces sperm. And firstly, to explain meiosis cell division or meiotic cell division or reduction division, first of all, we have to know that in the beginning, we start with interphase also, like mitosis. Chromosomes are double once in the interphase. But the difference here that mitosis is different than meiosis. Meiosis occurs in two stages. First, meiotic division, and second, meiotic division. And in the first meiotic division, we have prophase 1, metaphase 1, second anaphase 1, and telophase 1. Second meiotic division, we have prophase 2, metaphase 2, anaphase 2, and telophase 2. Firstly, what happens in prophase 1 as what happened in prophase, with an extra thing that makes there is a difference, that makes difference between meiosis and mitosis. The similarities appears in chromatin reticulum that intensifies and appears in form of distinct chromosomes. Also, spindle fiber that appears and chromosomes get connected with a spindle fiber from centromere. from centromere. Also, each chromosome consists of two chromatids, as what happened in mitosis, linked together by centromere. The difference here is that chromosomes are arranged in homologous pairs, in form of pairs. Two chromosomes beside each other. For example, chromosome like that, and another chromosome like that. Each chromosome consists of two chromatids, so we have four chromatids, which we call it tetrad. And at the end of the prophase 1, nuclear membrane disappears as usual, and every two homologous chromosomes in the tetrad starts to move away from each other after finishing very important thing, which is crossing over phenomenon. What the crossing over phenomenon that makes difference between meiosis and mitosis? In the following video, you will see that chromosomes are arranged, as you see here, in form of homologous pairs. Each two chromosomes are arranged together to form tetrad. And then what's important thing is happening in this case is the crossing over phenomenon. As we see here, crossing over happen, which is exchange of some parts of the red chromosome with the blue chromosome to form two new chromosomes different from each other, where the formed new chromosomes are mixed between the red and blue chromosome, okay, to form new chromosomes with new arrangement. To form new chromosomes with new arrangement. Crossing over phenomena occurs at the end of prophase 1, where some parts of two inner chromatids of each tetrad are exchanged to produce new genetic arrangement. Its function is to distribute genes between the tetrad chromatids randomly in the gametes, and variation of genetic traits among the members of the same species. So it tries to make new genetic arrangement. We have, for example, this green chromosome, and we have also this red chromosome. But after crossing over phenomena, we have red, and it took part of the green chromosome, and green chromosome, and it took part of the red chromosome. So it will be new arrangement, new traits. It makes difference. In the following video, we will see that. As we see here, chromosomes are arranged, okay, in form of homologous pairs, and then the crossing over will happen. New arrangement will appear. New traits will appear also. So this is the difference between, and the most important difference between meiosis and mitosis. Mitosis gives you same chromosomes with the same arrangement to give the same traits, identical traits. But in case of meiosis, they are mixed to produce new arrangement with new traits. After that, we have another difference in metaphase 1. In metaphase 1, chromosomes, pairs, not chromosomes, are arranged in form of pairs as a cell equator. In the following video, you will notice that the chromosomes pairs are arranged at the equator, not only chromosome or not each chromosome on the same line, but in form of pairs. In case of metaphase, they are arranged like that. But in case of metaphase 1, they are arranged in this form. And then we have anaphase. In anaphase, it's different than in anaphase 1, it's different than anaphase. In anaphase 1, every two homologous chromosomes start to move away from each other, where spindle fibers shrink. One of the two chromosomes migrate towards the cell pole, one of them move to this cell pole, and other one move to the other cell pole. Each pole contains half number of chromosomes. Why? Because here, this is different than anaphase. Anaphase, the chromosome splits lengthwise, like that, from centromere. But actually, in case of anaphase 1, whole of the chromosome moved to the right pole or the left pole, one of the poles of the cell. So, in this case, all of the chromosomes not divided from its centromere, they are splitted, or they split into two groups. Half of them moves to the one of the poles, and the other one moves to the other pole. So each pole have only half number of chromosomes. In the following video, you will see that some of chromosomes, or half number of chromosomes, move to one of the cell poles, and other one move to the other side of the cell poles. Which means that each pole has only half. And that is very important difference between meiosis and mitosis. Then we have telophase 1. And as what happened in case of telophase, also it will happen in telophase 1. Telophase is the opposite, or it's an adverse changes to what happened in prophase. And also telophase 1 is an adverse changes to what happened in prophase 1. So in this phase, at each of cell poles, a nuclear membrane is formed again around the chromosomes. So we have two nuclei. Each one has half the original number of the chromosomes of the parent cell. Then the cell enters into second meiotic division. Then the cell enters into the second cell into the second meiotic division. In the following video, as you see, spindle fiber disappeared, nuclear membrane appeared again, and nucleolus, and each cell of them has half number of chromosomes of the parent cell. And then we will go to the second meiotic division. And the second meiotic division is typical to mitosis. So, each cell of the two cells which resulted from first meiotic division is divided in a way similar to mitosis division phases. In the final phase, telophase 2 of this division, 4 cells are produced, and each of them contains half number of chromosomes of parent cell. It contains original number of organism's chromosomes. The number of chromosomes remains constant as it is mitosis. When the male gamete which is produced from the male, so any cell produced here is its gamete, male gamete or female gamete. When it combines with the female gamete, and as we know that male gamete has half number of chromosomes and female gamete also half number of chromosomes, the zygote produced which is the baby has full number of chromosomes like parents. For example, in human, male gamete has 23 chromosomes, female gamete also 23 chromosomes, so the parent will have 46 chromosomes. This is prophase 2, nuclear membrane disappear, and nucleolus disappear, spindle fiber appear, so as you see here, so as we see in the video here, nuclear membrane disappear and nucleolus disappear, and the spindle fiber appear to be connected to the chromosome from centromere. In the following video, which is metaphase 2, you will notice that chromosomes are arranged like mitosis, like metaphase. On the equator, in anaphase 2, chromosomes splits from centromere to form two identical groups of chromatids, as we see in the video. And in telophase 2, two cells are divided into 4 cells. Nuclear membrane appears again with the same number of chromosomes that it's found in the beginning of the meiosis 2, so it will not be divided again. At the end, meiosis 1 divided one cell into 2 cells, and meiosis divided 2 cells into 4 cells with half number of chromosomes of the parent cell. Division doesn't occur in red blood cells. This is very important note, as it doesn't have a nucleus. Also nervous cell, as it doesn't have centrosome. And there is another thing also. Some cells are not divided in normal conditions, like liver cells. Liver cells have the ability to divide under certain circumstances, which is under normal conditions, livers don't make division, but in case of injury, its remaining part makes mitotic division to complete it again. And we can use that in liver transplantation. We can cut part of one of the healthy man, we can cut part of a healthy man liver and put it to another one which is patient and has a bad liver, and then the cut part will grow up to form new liver with this patient, and this lost part from this normal man will grow again to form new liver. Also, we have very important thing to know, which is we have what's called nanotechnology. Nanotechnology is used in cancer treatment. First of all, what's the meaning of cancer? Cancer is uncontrolled cell division causes tumor, which means that cell division happens without control, the brain doesn't know anything about it, and then it will grow up in non-uniform shape or non-uniform way to form a tumor. Tumor, which means that part of your body, this tumor, means that part of your body grow up in improper way. So you'll find tumor in your hand, for example, or other parts of the body. So tumor is mass resulted from uncontrolled cell division. For example, this is your hand. Suddenly you'll find after, and then you'll find that part of your hand grow up For example, this is your hand, okay, part of your hand. Tumor means that part of your hand grew up to be like that, which means that growing but not in a controlled way, not in uniform way, there is no harmony, so it will be mass resulted from uncontrolled cell division, which is ingrowing, which is growing in improper way. This tumor, which is cancer, can cause a lot of troubles. But Egyptian scientist, Dr. Mustafa Said, discovered a way to detect cancer cells. How can we detect it? He detected it by loading a protein. They have the ability, this protein have the ability to attach to the cancer cells secretions with the nanomolecules of gold. As we see here, nanomolecules of gold attach, this is the patient, attach to the cancer cell, and just it attaches to it, we know that there is cancer in this part and we have to get rid of it. Here we have cancer in the brain. How can we get rid of it? To get rid of it, we have smart microscopic bomb. It penetrates cancer cell and explodes them from inside. And also we have laser. The idea of laser is the injection of the nanogold molecules that's surrounded by material that have the ability to attach to the cancer cell secretions, and then the infected cell surface and then the infected cell surface and protein get intertwined with golden molecules to make it possible to monitor the infected cell through microscope, each cell separately. So first thing, we can monitor each cancer cell. After that, by treating this way by laser with a certain degree will be focused on the gold molecule which is attached to the cancer cell, and then this cancer cell and then the gold molecule will take light and convert it into heat, which burn cancer cells so we can get rid of it. So we will use laser and focus it on the gold molecule that absorb light, convert it into heat, burn and kill the infected cell. As you see here, we are focusing laser on the nanogold molecules which turns light into heat and burn the cancer cell, and in this case we get rid of the cancer cells, which makes uncontrolled cell division, which is very bad because all of the body works in harmony and growing is not a mess, is not a chaos property. It's a way of organized growth in the body to form a body with a uniform and harmony, to form a body with a harmony in the growth and with a beautiful shape. But in case of the non-uniform or uncontrolled cell division, parts of the body, for example parts of the brain, will grow without control to form a tumor, and which will cause a lot of disease. One of them that it can, for example, which causes a lot of diseases. They were used to kill the cancer cells in an experimental mice suffered from cancer were able to live 300 days after this treatment. Other mice that didn't receive this treatment didn't live more than 43 days, and that's very good. As we see here, laser is focused on the cancer cells, and nanoparticles heat up, which creates tiny bubbles inside the cancer cells. The tiny bubbles quickly expand and burst, and the cancer cell rip the cancer cell apart. As you see here, it exploded or burned.